information, and the purpose for the jury and the judge is to see it's basically the same thing. Almost nothing's changed. There, on the left, you see there's more DNA present. The laboratory does the same experiment. It extracts the DNA, amplifies with PCR, and then separates and detects on a genetic analyzer. And now, what's the data we're seeing? Well, suppose there's another person here, and now we see, in addition to the 10 and the 12, there's an 11 allele. This suggests more than one person. The inference that we're talking about on the top right arrow is separating the DNA. Suppose, for the purpose of teaching, we know that the first genotype, the genotype of the first contributor at this locus, is a 10-12. This is the kind of question the computer's asking when it's solving a problem, by doing a lot of conditioning on other variables. What's the genotype of the second contributor? Well, we have to explain the 11. What can we explain it with? Not with an 8 or a 9, they're not on the slide. You don't see them in the data. We can explain it by pairing that new 11 allele with a 10, 11, or 12. So in a simple world, you'd have 3 possibilities shown on the top right: a 10, 11, 11, 11, 11, 12, and they have probabilities. It's for one contributor at a locus, just like we saw with the reference sample, but now there's probabilities that are not 100% attached to multiple possibilities. We'll also explain why the 10, 12 may be more probable, because if you add a 10, 12 with an 11, 12, you get 2 doses of 12, so there's more DNA. So we explain it. We adapt to juries, by the way. We don't memorize speeches. Some juries get everything in half an hour, other juries, they take an hour. We look at the jury, see what they're understanding. So does the lawyer. So this is what becomes different. Computers can use all the data. This is what data looks like in this case for one of the 75 evidence items at the BWA locus. The x-axis is showing numbers that, like 166, 171, this is how many base pairs are in the DNA, the length, the number of letters in the DNA paragraph. The numbers on top is the number of repeats. Once you account for the flanking regions, you just count up the number of repeated segments: 15, 16, 17, 18. The y-axis is the relative fluorescent units from genetic analyzer. It's low-level data. It's not up at 1,000. Some of the peaks are under 100. And so what do we see in data like this? First, we see heights of peaks. We don't see threshold of data. We don't see a list that says 15 and 17. Those 2 numbers are high enough. We don't do that. Our TrueAllele analyzes the HID file. It tells us what the allele, actually all events are at every base pair, and then it analyzes them. It needs to know the heights of the peaks. It will look at the pattern. It's trying to explain the pattern. And the main thing that it can look at that people cannot look at is the variation in the peaks. When the computer is comparing proposed patterns that explain data, you'll see one in a second, against the data, so it hypothesizes, what if I have certain contributors, I compare with the data, it's looking at the variation between what it's proposing as a model and what it's seeing in the data, and from those discrepancies, it can work out the variation. The variation is what you need to calculate an accurate probability. If you think of any probability formula you've seen, it's something like a difference in values over a standard deviation, right? E to the minus root. You need a baseline. You need variation. So TrueAllele has to measure the variation in the data to get accurate probabilities. In this case, people apply thresholds. They got less information. You'll see how. We'll show how the computer thinks to a jury. We do it by adding together individuals. Suppose, to explain this data, there's a person with a 15-17 genotype. So we have a certain amount of 15-17. You see those blue rectangles. Those are the 2 alleles. We represent quantity by the height. So maybe we have half as much, shown in orange, of a second genotype, a 16-17. Those start adding up together. And then there's a third person, maybe a 16, 18, showing green. So what's happening? The computer is proposing different amounts of different genotypes. It's going to test its proposal against the data. Here's another one. So maybe a fourth contributor, shown in red, is a 15, 18. The computer will try out tens of thousands of different patterns, putting patterns where the peaks are, where they aren't, correct heights, wrong heights. It's searching. It's trying to explain. Good explanations, where when you add together the alleles of the genotypes, you are getting the same pattern as the peak heights, will give more probability, more likelihood to those explanations. And most explanations are very bad. They don't put the genotypes where the data are, and they end up with very low likelihoods. There's a large separation between where the explanation is and where the data is, and so that will give lower probabilities. I guess I would love for half of you to go into this and tell you about the math, and it's in papers, and I can do that afterwards, but I don't have time, and half the room wouldn't understand. You can imagine I'd love to talk about that. But let me tell you about the case. When the computer has finished looking at doing these calculations of good explanations, bad explanations, for all 4 contributors at all 15 loci, that's 4 times 15, or 60 genotypes, 1 for each contributor at each locus. That's what we'll get at the end when we finish the Markov chain Monte Carlo. This is one of those 60 genotypes that the computer writes onto its hard drive and finishes. And this is what a probability genotype looks like. You've seen them before when you look at paternity. The original papers 100 years ago talked in this language, a probability of genotypes. You've seen this when you've looked at genotypes formed from population alleles. They have probability, kinship properties. What may be new to you is seeing probability for evidence genotypes from phenotypic data, like mixtures. But the scientists have seen these concepts before in other contexts. Of the 100 or so possible allele pairs, the allele pairs are shown on the bottom, the data for this contributor has concentrated probability on 8 or 10 possible allele pairs. It's increased the probability, as shown in the blue bars. The y-axis is probability, x-axis is allele pairs. For these allele pairs, it's reduced the probability for other allele pairs relative to a random person. Probability is a zero-sum game. In the end, it has to add up to 1. So where is the data constraining the genotypes to put their probability? This, by the way, never saw a comparison reference. It's not seeing the victims. It's not seeing the suspect. It's just separating the data into these probability genotypes. Now we want match information. So far, there's no person involved for a comparison. This is all being done objectively by a computer. The question we want to answer, which is one of about 10 ways of writing down the likelihood ratio with Bayes theorem, is, this is the most understandable one. How did the computers looking at the evidence data change the probability from a random person, shown by the brown bars on the bottom, that is, think of allele pair frequencies, to the probabilities we see, shown in blue, after the computers examined the evidence? How has probability changed? How is it increased? How is it decreased? If we take a look all the way on the left at 1616, you see that the brown probability before it looked at the data is higher than the 2% afterwards. The probability decreased. This would give a likelihood ratio less than 1. That would point away from a person with a 1660. On the other hand, the 1517 shows a large increase from random to evidence. I don't know, maybe it's tenfold. That would be the likelihood ratio. And when there's little change, if the blue and brown, the population probability and the evidence genotype probability are about the same, then mathematically there's little information, because the ratio is 1. The log of 1 is 0. I say to this half of the room, not the lawyers. And so that means 0 information. Still haven't made a comparison. All I'm showing you is now you can choose any reference, any genotype, even if it's not a person who exists, just any genotype, and now we can make a comparison. The computer has the probabilities of all the other genotypes written down. I'm just showing you the most probable ones. So we make a comparison with the victim. It turns out that at this VWA locus, at 1517, the victim happens to have 1517. That's what we didn't know. Now it's the first time, actually it can be done on a different computer or on the database. There are many ways of making a comparison in TrueAllele. Now we're going to take the ratio of the probability of an evidence match, that 48% in blue, divided by the probability of a coincidental match, which is about 16%. This is the easiest way to explain a likelihood ratio to courts. There are many hard ways, like the one I started with at the beginning, with Alan Turing and these strange conditional probabilities. This is the change in probability. That factor of 8, going from 6 to 48, is the match statistic. It's showing the change in probability. That's done at all 15 of these locations. The one we just looked at is on the left. Some numbers, here's the likelihood ratio on the bottom x-axis. Here are the 15 loci listed. Some have large numbers, some have small numbers. The one we looked at just now is in between. And because these loci are genetically independent, we can multiply them together. And that product of those 15 numbers tells us to what extent is the victim in the evidence. And the English that we can say is that a match between the bag knot and the victim, Apollo Torres, I'll look for the smallest number, is down under Hispanic, third one down, is 5 trillion times more probable than coincidence. So think of what it's saying. It's looking at the ratio of the change in probability. There's no mention of conditional probability. There's no mention of likelihood. There's no concept of that. It's what people understand. How do the probabilities change? By looking at the data. How do the probability of match change? Well, a match between the bag knot and the victim is 5 trillion times more probable than coincidence. That's a starting point for scientists and lawyers to begin to explain. It's just like random match probability, except that the numerator isn't 100%. It's less, at 50%, 40%. Otherwise, because the computer is separated into different contributors, we're only doing one contributor versus one contributor at the end. It's just like a simple single-source DNA in concept. We can explain it with pictures and words. There's brother, same concept of how we can calculate his statistic. Now getting more into the case. There was a lot of data in this case. Fortunately, our associate at Cybergenetics, Jennifer Brancamontes, was out there for days going through all 75 evidence items with pictures and statistics for all the references. TrueAllele found 4 or 5 other people's DNA that the crime lab didn't detect. We don't know who they are, but we know there are other people whose DNA was there. And we also compared different methods. I'll try to capture that for you for this 1 out of 75 items for everything that was compared in just 2 slides. You can imagine how the jury saw it. So this is brief by comparison. So this is a picture of areas near the garbage bag found outside the house that contained clothing. And the genotype, or the different genotypes that were separated from this 3 or 4 person mixture, are compared in the left column against 6 known references. We could have used negative match statistics. A negative 3 is 1 in 1,000. That's exclusionary. A negative 6 is 1 in 1,000,000. The lawyer felt this was so complex, just indicate that they're not included. Probably a good move, or there would have been too many numbers and heads would have started exploding in the jury box. So I'm not showing you the exclusionary numbers. We weren't asked to show them. So you'll see an X here. So in the first column, we see that these three individuals, including the defendant, are not present. We see that the brother is present with 4 zeros after the 1, a number in the tens of thousands, and that the victim, Apollo, is present with a match statistic of a log likelihood ratio of 12, which is a 1 followed by 12 zeros, or in American English, a trillion. What about these other references? These references were found by Trulio in cybernetics, not by the laboratory. And what we're finding, things that the lab couldn't see with their human review, is that there are matches in the hundreds and thousands, 2 zeros and 3 zeros, against these other two individuals. So this is showing all 10 comparisons, the 6 knowns and the 4 unknowns, in Trulio for this one evidence item. We then separately compared different labs. Originally, the prosecutors contacted Cybergenetics. Those are the results I just showed you. KRCL is the Kern Regional Crime Laboratory. They're a very good customer of ours. We do new technology with them. They're presenting papers on NGS and new technologies that we work with them on. The crime lab in the other county asked them to use Trulio, which usually they would do that, but they weren't sure what to do. And so the crime lab at the time didn't have error rates in place, so they weren't reporting on all their evidence. I think now they are. But where they did report, their results agreed with ours. This is the state lab in Santa Clara County. SCCCL is the Santa Clara County Crime Lab. You'll see they found nothing. Their inclusion methods couldn't say anything, even about the most informative evidence. That's what this case really hinged on, was the different worldview of primitive method that couldn't find anything versus computer methods. I'm just stressing this here, but our work was done independently by one of our customers, and we got the same results. This is the non-contributor analysis that I described, where we can compare against all possible genotypes of everybody on Earth and calculate error rates. The error rate for this match statistic of 14,000 was 191,000 people would match as strongly. This is a lovely paper. I still enjoy reading it. I go back to it when I need to understand what I did or built or what we're doing. It's published in Helion in 2018. There's a lot of words and pictures, and for people who like it, there's a lot of math. If you understand random variables and you will like DNA, you will probably love this paper. If you don't know what a random variable is, don't read the math. That's all I can say. But it lets us calculate error rates instantly, which we needed for 75 cases. So, let's get into what the lawyers are thinking. There are two different views of this. The prosecution is making the case that the defendant raped and killed a 2-year-old boy who lived in his house. That's what they're arguing. By the way, because they believe their crime lab is excellent, they never wavered from this belief. They didn't know what happened when they lost the case. They thought everybody made a mistake. They weren't listening to the evidence. They were listening to their crime lab, and their crime lab was wrong. The defense is saying, no, there's an abused toddler. It turns out there's a long history of abuse of the mother abusing and beating the child. Many visits to pediatricians, broken bruises. The mother is very abusive. And so there's an abused child who died, and the defendant had nothing to do with his death. We do have the evidence to explain. The prosecution said, look, the defendant's hair is on the clothing bag. On the left, we see the defendant. On the right, we see the evidence. Don't these look the same? Well, DNA analysis, if you look at the red square, showed they may look the same, but they're not. In fact, it's not his hair. Great forensic evidence under a microscope, but the DNA that we could find with Trulio is showing it's not him. Lab didn't see that. They just called it inconclusive. Inconclusive can lead to an injustice. This was a puzzle. The So there are pathology reports, first in the hospital and then an autopsy. And if you think about this puzzle, the first examination, the report said that there's the defendant's semen in the victim's rectum weakly positive in the hospital. And yet shortly thereafter, at the autopsy, it's completely negative. Why? What's going on? How does a positive semen turn into a negative semen? And in the hospital, Trulio agreed with the PATH report. There's a match statistic of 3.81, 3 zeros after 1. It's in the thousands. We agree. His DNA is weak. It's there. But at the autopsy, he's not there. So the DNA is agreeing with the microscopic, Trulio's DNA is agreeing with the microscopic findings of the pathologist. We don't know how that happened, but that's a big question for us. Why? That's one of our two big questions. By asking these forensic questions, it led us to what actually happened. There was sperm from the defendant on the victim's penis. Was it there? Well, the crime lab said, we see one cell. Must be there. Trulio analysis said, there's nothing there that's worth reporting. There's an exclusionary statistic. With one cell, it might be weak, but he's not there. So this is what happened. The crime laboratory looked at 97 evidence items. They reported 43 matches and one unknown person. We reprocessed 77 of the items that we were asked to, using Trulio. They're identical data. We reported 138 matches. 138 is maybe 3 times bigger than 43, what the crime lab found. We discovered 5 unknown people, the one the lab found, and 4 other people. I'm not going to go into this. This is a whole other talk about confirmation bias. There's another 30 slides that go with this. I gave it at American Academy meeting in a special talk 2 years ago. Not for today. It's just fun. I want to get on with the story. Basically, there was confirmation bias in what the lab was doing. They knew the answer. The computer did not have confirmation bias. It didn't know what anybody was looking for. It was just doing calculations. I'm going to show you some pictures. Each picture is 1/3 of a table. 75 evidence items. 25, 25, and 25. The darker the color, the stronger the match. Going all the way from just saying it's an inclusion in gray on the bottom to light blue, that's up to 1,000,000, 6 zeros, darker blue, 1,000,000,000, until you're finally getting to basically single source levels with very dark blue. The first 3 charts are the manual review of the crime lab. The next 3 charts are what Trulio found. This is what the crime lab found. They found inclusions to the victim. They didn't report statistics. And they see the defendant. They don't see the mother. This is going to become important. The mother's the primary caregiver, both by history and testimony. She's not there. Next, we see more of the victim in the center. We see the defendant in the red column. There's some more matches against evidence items. The mother is not there. The manual methods are not seeing anything else. Here's the victim who reports the match statistics, and then the crime lab on these last 25 items is reporting inclusions and some match statistics to the defendant. I think that might be as an unknown, it's finding something, and nothing to the mother. So it's, imagine you look at the sky and all you see are clouds and a few lights. That's what you're seeing. With Trulio, the sky lights up with stars. You're seeing what happened. It's looking into the evidence. It's not throwing out. It's not doing anything. It's telling you what's there. We're seeing on the left match to the brother, which we might expect by cross imaging. We're seeing the defendant. We're seeing more of the defendant than the crime lab does, by a lot. We're seeing more of the victim. We're seeing these other individuals unknown to the crime lab are matching, some of them very strongly. This is the first 25. We don't see the mother. Even Trulio isn't seeing the mother, the caretaker. We continue with the next 25 items. We see the victim, strong match statistics. The individuals the crime lab isn't seeing, there are matches to. These are other people. The evidence is almost pointing more, if you're going just on counting matches, to the people we don't know than to the defendant. Still no mother. And it continues with the last 25. There's the victim, everything's based on match statistics. We see other individuals. We see the defendant in the red column, and no mother. But that's really a question. How can someone be the primary caretaker of a child, and there's none of her DNA anywhere from this crime scene on the child, on objects, nothing? I mean, she says she bathed him, no DNA. Does she not have DNA? What is she a robot? I don't know. We'll get to it. So here's our two puzzles. And I'll tell you the bravery of the senior public defender, Kelly Kulik, who became a judge, is she's seeing far more matches to her defendant with Trulio than with the lab. And she's not fazed. She's saying, the science, that's the science. Now explain it to me. He's not guilty. We know he's just not guilty. So figure out what happened, because if you don't figure out what happened from all this DNA, he may be executed. Here's the first puzzle. Where's the mother's DNA? It's nobody's understanding this who's looking at the case. Lots of different people left so much DNA. There are so many matches, but the primary caretaker has no detectable DNA. The rectal DNA complex. How could the defendant's DNA be found in the initial hospital swab, but not soon after the autopsy? It's supposed to be inside the body. What's going on? Two different views of the crime. They're looking at the same DNA evidence. They're reaching different conclusions. The prosecution raped and killed the child who lived in his house. The defense, this is an abused toddler, he died. The forensic evidence isn't relevant. The problem with this is each of the lawyers are assuming their side, and their conclusion is what they've assumed. There's no science here, they're just arguing, right? My client is innocent, therefore he's innocent. Your client is guilty, therefore he's guilty. There's no real argument involving forensics. That's what scientists are supposed to do. To see it from their view and the way they present it in trial, the prosecution sees crime, the forensics, the DNA, all these objects, DNA is everywhere, he must be guilty. There's no explanation, but he must be guilty. The defense sees nothing criminal. This is a quote from the closing argument, which I think was 6 hours. It's trying to explain everything to the jury. Lots of people's DNA was left in a messy, dirty, spermy house. That's her conclusion. So what does the science tell us? All right, we find some transcripts. I'm asking the defender questions, and so what happened with the mother's DNA? And we come across this transcript, and basically, in this interview with the police, the mother says that she had oral sex with her boyfriend, and she had DNA all over her hands, and she cleaned it. His semen was all over her hands, and she cleaned it up with some baby wipes. Completely new information, right? And then she threw them out. All right. Well, if you take a person whose DNA is not seen at a crime scene, who's touching the baby and everything around continuously, and she has somebody's semen on her hands, and she's touching everything, then one, you're not going to see her DNA, and two, you're going to see her boyfriend's DNA on everything. Because it doesn't mean anything. We now know there's massive contamination. Whatever DNA anybody had is completely masked by the DNA from the sperm on the mother's hand, who's touching everything. So, therefore, to the lawyers, I'd say, there's no probative value in the DNA because we now know that it can't tell us anything. The amount of DNA from the defendant's sperm is so much more than all this touch DNA. We're just seeing it everywhere. And we know it's true because we're not seeing the mother's DNA anywhere. And by her testimony, she's touched everything. So she's leaving the defendant's DNA on everything. Not intentionally. Doesn't have a science background, but that's what it is. What about the rectal DNA conflict? Again, a few days before I testified, we're looking through transcripts. This is during COVID, so Kelly's in California, I'm in Pittsburgh, we're having Zoom meetings and trying to figure it out. And then we read how the swabbing is done at the hospital. This is an interview with the pathologist. And the description is that, in great detail, I'll summarize it, you can read it, is that the pathologist is taking a swab in sterile water, swabbing the outside of the anal region, and then just going in a little bit to sample the rectal region. And it's called a rectal swab. What's the problem? The problem is language. This is an anal rectal swab. They're first cleaning up an area which, if it's contaminated by the defendant's semen, is outside. But they're reporting it, calling it rectal, as if it's inside. And so, this was more complex to explain to the jury. They got it. That the mother transferred the defendant's semen from her hands onto the bottom of the toddler. And that's what they're seeing the first time at the hospital. The first pathologist is swabbing the anus on the outside, cleaning it, getting all the semen from the mother, from the boyfriend, onto the swab. And then you've completely contaminated by cleaning up the rectal swab, but you're not calling it a rectal swab. You're calling it a rectal swab, but it's really cleaning up the outside area. And you're reporting it as if it's just the inside area, which is leading to the criminal charge of rape and sexual assault. Because the prosecution is thinking the only way that his semen could have entered the child is through some forcible rape. But it's being collected from the outside, transferred from the mother on her hands. And at the autopsy, the first pathologist had already cleaned up the bottom, so there's no semen left. And there's no semen inside either, so now it's negative. But these were the two facts that were very hard to explain. And we could explain them by the testimony of the mother and the pathologists. This is what they did. This is why the surprise went away. First pathologist cleaned up the outside, nothing left for the second pathologist at autopsy. They used the words rectal swab. The jury's ready to execute this man. Horrible crime. But it's just words. So the jury, having listened to this, acquitted the defendant of all charges. And the prosecutor, the head prosecutor, the district attorney, was so surprised by the outcome, they actually eliminated the death penalty in Santa Clara County after this case. Because the concept that the evidence might have misled them and they would have executed somebody innocent was pretty horrifying. Ultimately, the mother was convicted of something related to the death. Okay, so the issue is, oh, this is great. Not only do I have another half an hour, but you can't leave. So, I'm going to tell you about another case, but there aren't that many more slides. I hope I've left some time for questions. This is more typical. This is a prosecution case. This is a double homicide that we worked on 10 years ago in Pittsburgh. And it's much more typical. Usually the DNA evidence favors the prosecution in the nature of crime. It sometimes favors the defense or innocence. But this story has a lot of lessons. So we begin by saying, with human review, crime labs don't use all the DNA and apply thresholds. The data just isn't all used. This is the Wolf sisters homicide. In 2014, two sisters who came from Iowa moved to the Friendship neighborhood of Pittsburgh, Susan Wolf and Sarah Wolf, and they had a home in East Liberty, and they were found killed. There was no motive, no known motive, nobody knew what happened. That's not typical for Pittsburgh that women are found dead in their homes. It's not a thing. The crime lab looked at, I don't know, maybe 16 evidence items, but their protocols didn't let them use all the DNA data. They really can't handle complex DNA evidence. They applied thresholds, and what they were able to say for most of the evidence is that they couldn't interpret it. They gave many reasons, and you may have seen language like this if you have it in your report. If you see American reports, you see language like this. There was a hat, no conclusions. This is summarizing many pages of reports. There was a cup, insufficient data. DNA under the fingernails of the victim was contaminated, insufficient data. A gear shift in a car, video footage caught somebody driving away. Insufficient data. The seat lever cannot be excluded. The weak match statistic doesn't mean much. There was the knit hat that I'll come back to later. Insufficient data. There was the sock, too complex, no conclusions. Do you see language like this in some reports? Or people just don't bother writing it down? In America, you have to write down what you didn't find in your report. So this is what they didn't find. TrueAllele can use all the data. You see how it tries to explain the patterns, it measures the variation, it works out probabilities, it tries to explain the data tens of thousands of times at every locus, adding together different genotypes that explain the data well or explain it poorly, better explanations, where the data pattern that you see in front of you, of middle, high, low, low at this one locus, looks like the explanation pattern of middle, high, low, low. There's the match. All right. The math is, the computer doesn't know about a match. It knows mathematically they're close. It can calculate that the heights of the peaks are close to the heights of the rectangles. And then we get an evidence genotype at each of these. I think it's also 15 loci with 3 contributors, each of the 45 loci. This is what the jury sees. We explain the computer concentrates the probability on these 4 allele pairs out of the 100, and then we can make a comparison with the brown bars, which is from population allele frequencies, what are the probabilities of these genotypes, and then we can get ratios like 50% to 5%. It happens to be that if the defendant had a different genotype, it would have been a different number. If he happened to be a 15-16, like we see on the left, it would have been exclusionary. Coincidence had more probability than evidence match. But when we compared with him, the computer said, well, 16-16 genotype is locus, 55% divided by 5%, that's the change in probability, that's the likelihood ratio. Now we look at the match information at these 15 locations, the same chart. This is the likelihood ratio on the x-axis, the 15 loci. Again, when we report, we don't want to, if we can, give the largest to the smallest. We always like talking about typical or average. What's representative? And so here, you see that the VWA statistic of around 10 is typical. It's not, over 100, it's not. It's just kind of in the middle. And now we can ask, based on the match statistic, not on what we see, but what the computer has calculated, to what extent is the defendant in the evidence. And we can state to the jury and explain further if asked, we've already shown the picture, that a match, see, I have to tell you, you see here the phrase more probable, it's in the middle of the sentence, right? More probable. It took years to come up with language like this. The purpose of using the word more probable is if you don't put that phrase in there, suddenly you're talking about the probability of a match, and people get confused. But if you say something is more probable than something else, then you're talking about probabilities, but you're not hurting the minds of the jurors or the judge. You're saying it's more probable, but you're not using so many words that have math with them that jurors don't want to hear you. you are saying it. It's more probable that there was sunshine this morning than rain. You could expand that into the probability of sunshine today was greater than the probability of rain. You said the same thing. You've conveyed the same idea, but it's easier for people who aren't scientists to hear more probably. It's that kind of being careful with language. But this is reading off the math. It's one of the forms of the likelihood ratio. A match between the fingernails and the defendant is 6 trillion times more probable than coincidence. Okay, that's as simple as we can get it. I don't know if the list of references, articles was distributed. That's one of the 4 articles that I'd sent out that you might want to read if you're interested in how do you explain the likelihood ratio. I think I wrote it 15 years ago. This is what we found. The crime lab could report 5 DNA mixture matches. Most of them weren't very probative. TrueAllele found 17 matches on the same data. The hat was 65,000 to Alan Wade, cup 20,000 to Susan Wolf. The DNA under the fingernails, 6 trillion to the defendant, Alan Wade. DNA under fingernails is very probative. How is DNA from a defendant getting under the fingernails of somebody who's dead? It's very hard to explain in court. If they say they didn't talk, they didn't eat, they didn't do anything, there's the DNA. So that's the probative power of mixtures, is that 2 people's DNA is together someplace it's not supposed to be. Whether it's a sexual assault or a homicide, why is the rapist's DNA in the rape kit? It's not supposed to be there. They never met her. How could it be there? It's the most powerful DNA evidence is usually mixtures. It explains what happens. It corroborates, supports the statements that witnesses make about what happened. Anyway, so we continue. Lots of match statistics. At the trial, there was so much evidence. I didn't watch the closing argument, but the prosecutor put in time sequence each of the items that we found the DNA match to in front of the jury box. And then I was just told about it. And he walked through the story in time, walking with each piece of evidence inexorably leading to the defendant. That's a good moment for a lawyer. It doesn't affect scientists, but he had so much evidence that he could tell the story supported by bank cards, video cameras. There it was. There was the DNA. And with the older method, he had basically nothing. So he was convicted, found guilty of first-degree murder. But there's another moral to the story, which is that, as you can expect, if a crime lab knows that it cannot interpret certain evidence, in the interest of efficiency, it's probably not even going to test it. If someone brings in a doorknob from a crime scene, we've done doorknobs. They've been used to help convict people. There's 5 or 6 people mixed together on a typical doorknob. If a laboratory is struggling to do 2-person mixtures, they're going to say, no doorknobs. We're not going to analyze them. Handguns typically have 5 or 6 contributors. We get a lot of handguns from, especially federal prosecutors. They handle a lot of handgun cases. The crime lab says it's inconclusive, but at least we have the data, we separate it out, and we can help the justice system with those. Hats were something that this crime lab didn't accept, generally. So the detectives knew to not even submit a hat, because the crime lab wouldn't analyze it. They also had a backlog of half a year or something, but there's too many people. A hat. Criminals share hats. Social life. There's a lot of DNA in the hats. So what actually happened is that Alan Wade was the next door neighbor of the Wolf sisters in Friendship. And there had been a burglary of the Wolf sisters' home 6 weeks before the murder. And at that burglary, a hat was found. Now, at the time, the police didn't submit the hat. They knew the crime lab wouldn't analyze it. And the crime lab wouldn't have analyzed arrested because they don't analyze hats. But after the murder, 6 weeks later, the district attorney's office said, you're dropping everything else in your lab. We need evidence now from everything. And so they did. Everything was done just on this one case, and they produced all this evidence, most of which they were correct, they couldn't interpret, but Trulio can. But ask yourself a question. If DNA could be processed automatically and quickly with each step of the process, taking a few hours to extract, maybe one day using robots to PCR, separate, interpret, and in a few weeks, all DNA evidence had been analyzed by this lab, including the hat from this burglary. They would have known that Alan Wade was burglarizing the house. And the district attorney told me that he felt if that had been known, then the sisters never would have been killed, because the murderer would have been detected as a burglar a month before. So that's the danger of not fully analyzing DNA evidence. When it becomes inconclusive, evidence that can be used to prevent crime isn't, and crime continues. Think of serial rapists and so on, serial criminals. So I'll just say, in conclusion, I'll show 2 slides. Typically, there's no information from complex mixtures. In America, handguns largely go unanalyzed. They may have 4, 5, 7 people on them. Here's typical result. It's a lot of words that says we can reach no conclusions. So government does whatever it does, there's no conclusions. This case was interesting in that we got it and never went to trial. Most of our cases don't go to trial because in America, once there's enough physical evidence, defendants will plead guilty. It's a different story, another lecture. You can have entire symposia on them with lawyers, but that's what happened. So what happened in this case, I was told about this both by the prosecutor and by the defender, because I was auditing a law class that he was teaching, and he let me audit the class in part because it helped his client. In part. I'm sure he would have let me audit it anyway. It was on criminal procedure. So Trulio took the same DNA evidence from a handgun. Well, that's fine. It's a great story. There's some teenagers driving around town. Shots are exchanged. There's a policeman walking by. He thinks they're shooting at him. Finally, they arrest people. They find a gun. Everybody says they didn't touch it. Right? Otherwise, we wouldn't be here doing DNA. Oh, I touched it. No, but they all said they didn't touch it. They unmix. Cybernetics unmixes this DNA mixture from this inconclusive result. We find that there are 4 contributors. Person A is excluded. Oh, now we have information that charges are dropped against that person. There's no point in taking this person to trial if the computer has shown he's not on the gun. Okay, that's good. For another contributor, there's a match statistic of about 500,000 to a second person. Well, when he's confronted with the DNA report, he confesses that he handled the gun. Gets a lesser sentence, there's no trial, life goes on. So by having the information, the right people are being connected to the right crimes, and the people who are not connected are not being connected, and it's being resolved with physical evidence. It's not going to court with people arguing about evidence that's not that informative. Trulio today. So the math, the algorithms, they're over 25 years old when I developed them. Trulio is a computer system as we know it now. We started developing as a Bayesian probability system over 20 years ago. We have 10 laboratories who use it regularly. They've done, it's 100,000 cases yet, but certainly many tens of thousands of cases. In cybernetics consulting work, the laboratories use Trulio in their own lab, and other groups come to us. When software doesn't work in a laboratory, usually the prosecutor or the defender will come to cybernetics, often the police. And we counted it up recently, and we've worked for 400 agencies of all types: police, prosecutors, innocence groups, offenders, and so on. I won't, the innocence cases are great, but I'm out of time. They're wonderful cases to work on. On our webpage, we describe the 11 exonerations we've done so far for wrongful convictions. There have been 42 validation studies. We've given hundreds of talks. I checked with our office today and asked them to count it up. We've educated over 800 students, mainly forensic science students, about Trulio. There have been almost 40 rulings across 5 states in the U.S. and foreign countries and federal courts. Cybergenetics has testified in over 100 trials, and we've worked on over 1,000 cases. So I think time is good. A lot of information that you can get from our website or from asking me questions or meeting later on. I appreciate your hospitality. It's been wonderful. I can answer questions now, more later, whatever you like.